Good day everyone. In this chapter, we will discuss about the DNA structure, analysis, and the replication. To understand this topic, we have some key informations as to what we can expect to understand in this chapter. First, the structure of DNA provides the basis for storing and expressing genetic information. Second, RNA has many similarities to DNA but exists mostly as a single-stranded molecule. Third, in some viruses, RNA serves as a genetic material. And last but certainly not the least, with the exception of some viruses, DNA serves as a genetic material in all living organisms on Earth. We will begin to understand all of these concepts as we go along in this lecture. First, let us begin by knowing and understanding the four characteristics a genetic material must exhibit. First is the replication, second is the storage of information, third is the expression of information, and last is variation by mutation. We will understand all these processes as we go along our lecture. But first, let us shift our focus in this figure. In this figure, it shows a simplified view of the information flow, which is also known as the central dogma involving the DNA, RNA, and the proteins. From the chart, we can see that genetic information flows in a specific direction. It goes from existing DNA to make new DNA, which is known as the process of replication. Then the DNA undergoes transcription, which produces mRNA, the messenger RNA, the rRNA, the ribosomal RNA protein synthesizing the RNA, and the tRNA, which is the transfer RNA, which helps decode messenger RNA sequence into a protein. These RNAs are transported out of the nucleus into the cytoplasm and then to the ribosome. Lastly, the ribosome undergoes the translation to produce the protein. First, we have the replication. The replication is the process when existing DNA is copied to make more DNA. DNA replication is semi-conservative. Now, what do we mean by semi-conservative? It just means that one of the strands of the DNA in each of the two copies of the DNA is considered ancient and conserved, while the other is newly produced. There are also three major steps in replication. First is the opening of the double helix and separation of the DNA strand. Second is priming of the template strand. And third is assembly of the new DNA segment. Let us try to understand the three major steps of replication through the DNA replication fork. This structure is a DNA helicase in which energy from the ATP, adenosine triphosphate, helps to continue unwinding the DNA, forming a structure called the replication fork. The unzipping of the two strands is the process of breaking the hydrogen bonds between the nucleotide base pairs in double-stranded DNA. New DNA is made by the enzymes called the DNA polymerases, which require a template and a primer, also known as our starter, and synthesize in the 5' to 3' direction. To understand the replication fork, first we will start with the 5' to 3' associated with the primer. DNA polymerase can only make DNA in the 5' to 3' direction, and with this, it creates a problem during replication. A DNA double helix is always anti-parallel, which means that they are opposite. One strand of the DNA runs in the 5' to 3' direction, while the other is 3' to 5' direction. But these differences are actually necessary so that the two new strands will also be anti-parallel to their templates to be made in slightly different ways. The one new strand, which runs in the 5' to 3' towards the replication fork, is the easy one, due to the fact that this strand is made continuously because the DNA polymerase is moving in the same direction as the replication fork. The continuously synthesized strand is called the leading strand. The other new strand, which runs to the 5' to 3' away from the fork, is considered trickier. This strand is made in fragments because as the fork moves forward, the DNA polymerase moves away from the fork. This must come off and reattach on the newly exposed DNA, and then this strand is called the lagging strand. The small fragments are called the Okazaki fragments. This term is named after the Japanese scientist who discovered them. The leading strand can be extended from one primer alone, whereas the lagging strand needs a new primer for each of the short Okazaki fragments. DNA replication requires other enzymes in addition to DNA polymerase, including the DNA primase, DNA helicase, DNA ligase, and the topoisomerase. We will understand this in the succeeding slide. First, we have the DNA primase. A primase's main purpose is to initiate the addition of new nitrogen-containing bases to the single-stranded DNA that was brought about by the actions of the helicase. A DNA primer provides a starting point for DNA synthesis. A primer must be synthesized by an enzyme called the primase. Next, we have the DNA helicase. Helicases are enzymes that bind and may even remodel nucleic acid or nucleic acid protein complexes. As mentioned from the replication, it is essential because it separates the double-stranded DNA into a single strand to allow the replication of both strands. Next, we have the DNA ligase. an enzyme that catalyzes the formation of a covalent bond between a 5' phosphate and a 3' hydroxyl group at the termini of the DNA fragments. This bond ligates the two DNA fragments together. DNA ligase could also be noted that it is used on the lagging strand because it is used to join the Okazaki fragments. Last, we have the topoisomerase. Topoisomerase are enzymes that catalyze changes in the topological state of DNA, interconverting relaxed and supercoiled forms, linked or decatenated and unlinked species, the knotted and the unknotted DNA. Topoisomerase is responsible for preventing the DNA from getting tangled as well as relieving pressure in supercoiled DNA during the replication. Next, we have transcription. Transcription is the process by which the information in a strand of DNA is copied into a new molecule of messenger RNA or the mRNA. This is also the storage of information. Transcription is also the process in which a cell makes an RNA copy of a gene's DNA sequence. Just like replication, transcription also has its three major steps. First, we have the initiation, in which RNA polymerase binds to a sequence of DNA called the promoter. This is found near the beginning of the gene. Second is the elongation, in which one strand of DNA, the template strand, acts as a template for RNA polymerase. And then last, you have the termination. Sequences called terminators signal that the RNA transcript is complete. Let us try to understand these three major steps one by one. Let us begin with initiation. RNA polymerase binds to a sequence of DNA called the promoter. This is found near the beginning of a gene. Each gene or group of co-transcribed genes in bacteria has its own promoter. Once bound, RNA polymerase separates the DNA strands, providing the single-stranded template needed for transcription. As you can see from the image here, the RNA polymerase is responsible for separating two single-stranded templates from the DNA. These two single-stranded templates will be used for transcription. Next, we have elongation. One strand of the DNA, the template strand, acts as a template for RNA polymerase. As it reads this template one base at a time, the polymerase builds an RNA molecule out of complementary nucleotides, making a chain that grows from 5' to 3'. The RNA transcript carries the same information as the non-template or the coding strand of DNA, but it contains the base uracil U instead of the thymine T. So looking at this image, we have the template strand, the RNA, and the coding strand. Let us shift our focus on the template strand, since this will be the most important one because it will serve as the basis for coding the RNA and also understanding the coding strand. So before we try to understand this, let us understand or put into idea that the pair of a thymine is adenine and the pair of a guanine is cytosine. So A is to T is to A, G is to C is to G. So to incorporate this idea with the template strand and the coding strand, as you can see in the template strand, TAC, TAG, AGC, and ATT, when it comes to coding strand, we will just replace the T with its complementary pair. Also, of course, the C with its complementary pair, the G, and vice versa. So looking at the template strand and the coding strand, T is to A is to T, C is to G, T is to A is to T, G is to C, A is to T, G is to C, C is to G, A is to T is to A, and T is to A. So we actually just replaced the complementary from the template strand to the coding strand. So now we move forward to the RNA. How do we code the RNA? It's actually easier if we have the coding strand. So looking at the coding strand, let us start with the ATG, ATC. When it comes to RNA, you should just replace the thymine with the uracil. So instead of retaining the T, you change it to U. So AUG, AUC. Looking at the coding strand ATG, ATC, we just replaced the T with the uracil. Now, how come we replace the thymine with uracil in the RNA? Uracil is actually less stable than the thymine, hence why we use the RNA, so that there are no much problems when it comes to synthesizing, since it will break down easier than the thymine. And lastly, we have the termination. Sequences called terminators signal that the RNA transcript is complete. Once they are transcribed, they cause the transcript to be released from the RNA polymerase. An example of a termination mechanism is involving the formation of a hairpin in the RNA. So the reason why there is hairpin formation, it regulates the transcription process and controls the synthesis of several amino acids including histidine, lysine, threonine, and phenylalanine. Next, we have the translation. Translation is the process through which information encoded in the messenger RNA directs the addition of amino acids during protein synthesis. Translation then decodes mRNA into amino acids, forming proteins essential for life functions. In terms with the three major steps in translation and transcription, they remain the same, but it could be noted that the difference between transcription and translation is that the process by which DNA is copied to RNA is called transcription. And that if RNA is used to produce proteins, then it is called the translation. And with this, let us proceed with the proteins. Mutation creates variations in protein coding portions of genes that can affect the protein itself. So, from the term itself, variations are caused by mutations. Protein modifications at amino acids or the polypeptide chain generate large numbers of variations.
Before continuing with the topic regarding genetic material, we must first go back to its history and understand how geneticists figure out that DNA holds a genetic material. Genetic material is physically transmitted from parent to offspring. This fundamental concept is crucial in genetics as it underlies the inheritance of traits from one generation to the next. For a long time, proteins and nucleic acids were the major candidates considered for genetic material due to their abundance and diversity in cells. So here we have the pioneers in the discovery of DNA as genetic material. First, we have Friedrich Miescher. He is a Swiss physician and biologist. He was the first to identify a substance he called nuclein, or now we know as a DNA. His work laid the foundation for the discovery of nucleic acids, although the significance of his discovery in relation to genetics was not realized at the time. Next, we have Phoebus Levine. Phoebus Levine is a Russian-American biochemist. He made a significant contribution to the understanding of the chemical nature of nucleic acids. In 1910, he proposed that DNA was composed of a series of four nucleotide units, each containing a sugar, a phosphate group, and an adenosine base. And then third, we have Erwin Chargaff. He is an Austrian-American biochemist. He conducted experiments in the 1940s that led to the discovery of the two key rules now we known as the Chargaff's rule and debunked Levin's hypotheses. So all throughout this PowerPoint, we will be talking about Chargaff's rules. Moving on, his findings were crucial for the later discovery of the double helix structure of DNA by Watson and Crick. These early discoveries set the stage for understanding that DNA, rather than proteins, is the molecule that carries genetic information. The journey from initial identification of nucleic acids to the realization of their role as genetic material was a critical chapter in the history of genetics. So now, we will be talking about the experiments which serve as evidence favoring DNA as the genetic material. So first, we have Griffith's experiment on transforming principle. So here he used two strains of the Diplococcus pneumoniae, or now we known as Streptococcus pneumoniae. So he has a strain of a virulent smooth with a polysaccharide coat that causes the disease, and a non-virulent rough R strain without the coat that does not cause the disease. He injected four different mice with four different samples. First is the live S strain where the mice died. Second is the live R strain, the mice survived. Third is a heat-killed S strain where the mice survive. And the last one is a mixture of heat-killed S strain and the live R strain where the mice died. He observed that the blood of the dead mice contained a live S strain bacteria, wherein he concluded that some transforming principle from the heat-killed S strain bacteria was taken up by the live R strain bacteria, transforming them into a virulent strain. This suggested that the genetic material responsible for virulence could be transferred between bacteria. It indicates that the presence of a molecule that carries genetic information. Next, we have Avery, MacLeod, and McCarthy's experiment. So they expanded on Griffith's work to identify the transforming principle. So here, they used a purified extract from the heat-killed Streptococcus pneumoniae, or the S strain, and then the extracted was treated with a virus enzymes to selectively destroy proteins, RNA, and DNA. So they used protease, ribonuclease, and deoxyribonuclease. They then mixed these treated extracts with the live or strain bacteria and observed whether the transformation into the virulent S strain occurred. So with the results, extracts treated with protease and ribonuclease still transformed the R strain bacteria into the virulent S strain, whereas the extracts treated with the oxyribonuclease did not cause transformation. They then concluded that DNA is a transforming principle and the molecule responsible for transferring genetic information, as only the destruction of DNA prevented transformation. This provided strong evidence that DNA, not protein, is a genetic material. Before we move on to the next experiment, which is the Percy and Chase experiment, we must first understand the T2 bacteriophage and its life cycle. So, the T2 bacteriophage is a type of virus that infects bacteria, specifically E. coli. It consists of a protein coat encapsulating its DNA. Here we see the life cycle of the T2 bacteriophage. First, we have the attachment. The bacteriophage attaches to the surface of the bacterial cell. Second, injection. The phage injects its DNA into the bacterial cell, leaving its protein coat outside. Third would be the replication. Inside the bacterial cell, the phage DNA directs synthesis of a new phage components using the bacterial machinery. Fourth would be the assembly. New phage particles are assembled into the synthesized components. And lastly, lysis. The bacterial cell bursts, releasing new phages, which can infect other bacterial cells. So, lastly, we have the Hershey-Chase experiment. The objective for this experiment is to determine whether the DNA or protein is the genetic material that phages transfer to bacteria. So, first, they labeled the phage components. Phages were grown in two different media to label their DNA and proteins separately. One batch of phages were grown in the presence of radioactive sulfur or 35S labeled proteins, since sulfur is in protein but not in DNA. Another batch was grown in the presence of radioactive phosphorus or 32P to label DNA, since again phosphorus is in DNA but not in proteins. Next is the inspection stage. So the labeled phages were used to inspect the E. coli bacteria. The mixture was allowed to proceed for a short period, enabling the phages to inject their genetic material into the bacteria. Then, the mixtures were then agitated in a blender to separate the phage protein coats from the bacterial cells. The samples were centrifuged to separate the heavier bacterial cells from the lighter phage coats. And then the bacterial cells and the supernatant containing the phage coats were separately analyzed for radioactivity. The results: bacteria infected with 32P labeled phages contain radioactive DNA, whereas the bacteria infected with 35S labeled phages did not contain radioactive protein. So, Hershey and Chase concluded that DNA, and not protein, is the genetic material that is injected into the bacteria by phages and directs the production of new phages. This provided definitive evidence that DNA is the molecule responsible for heredity.
So, under the direct evidence, recombinant DNA studies, recombinant DNA technology combines DNA sequences from different organisms. An example of this is human insulin gene, which is spliced into bacterial DNA, leading to insulin production in bacteria. Genomics revealed the full set of DNA sequences in organism, and studying genomes of individuals with heritable disorders identifies DNA sequences responsible for mutations. And overall, DNA is accepted as the genetic material in eukaryotes. Under indirect evidence, which is the distribution of DNA is found primarily in the nucleus as part of chromosomes. So, protein is abundant in the cytoplasm, but it is not specifically associated with its genetic function. Mitochondria and chloroplast performs genetic functions and also contains DNA. With this observation, it favors DNA over protein as the genetic material. So, indirect evidence, it is also where DNA was encoded to the human bee globin gene and was microinjected into a fertilized mouse egg. It is later found then to be present and expressed in adult mouse tissue, and it is transmitted and expressed in the mouse's progeny. This was conducted by Beatrice Means. With indirect evidence, this is where we can find the UV light's correlation to mutagenesis. So the UV light induces mutations in genetic material, and an action spectrum of UV light as mutagenic agent can be compared with the absorption spectrum of the suspected genetic material. So with DNA and RNA, it absorbs UV light at 260 nanometers, while with protein, it absorbs around 280 nanometers. So the absence of significant mutagenic effects at 280 nanometers supports that nucleic acid such as DNA or RNA is the genetic material. All right, so after these experiments, people were really believing that, okay, DNA is indeed a genetic material, but people don't really have a good background on how it was structured, how it replicates, how it stores information, and how it is passed on to the offspring. And so this is where Watson and Crick comes along during 1953. They used Rosalind Franklin's data of X-ray diffraction analysis, and they noted that DNA is a double helix structure. But during the Nobel Prize award, it was only Watson, Crick, and Wilkins that was recognized because Rosalind Franklin already died during the time. So in here you can see the nitrogen bases, which can either be purine or pyrimidine. And purine consists of adenine and guanine, while pyrimidine consists of cytosine and thymine. And in thymine, it can be uracil in RNA. Nucleotides are the building blocks of nucleic acid. And so nucleoside is a nitrogenous base and a pentyl sugar, but when you add phosphate group, it then becomes a nucleotide. And in DNA, pentyl sugar is deoxyribose sugar, while in RNA it is ribose sugar. Also, the glycosylic bond is found between sugar C1 and the base. And in phosphoester bonds, it is between the sugar C5 and phosphate. The backbone of DNA is created via phosphodiester bonds between sugar and phosphate groups on adjacent nucleotides. So the 5' phosphate group binds to the 3' hydroxyl group on the neighboring nucleotide. And this bond gives directionality to the linearity of DNA strand. The string of nucleotides is polar due to the charge of phosphate groups and the hydroxyl groups on the sugar. So in here, DNA double helix is created via bonds between two linear DNA strands. Sugar phosphate backbone forms the outside edges with the bases facing the base pairs. So there are 10 base pairs per helical turn. And one helical turn adds 3.4 nanometers to length of DNA, which is 0.34 nanometers per nucleotide pair. Two complementary strands run anti-parallel to make up the DNA helix, and the helix forms a major groove and minor.
For the Chargaff's rule, the number of purines and pyrimidines exists in the ratio 1:1. This is because the purines bond with pyrimidines to form nucleotides. Hence, in the double-stranded DNA, the number of purines is equal to the number of pyrimidines. But the percentage of A plus T is not necessarily equal to the percentage of C plus G. In the X-ray diffraction analysis, it is the process where the fibers of a DNA molecule are subjected to X-ray bombardment. This causes the X-rays to scatter according to the molecule's atomic structure, and the pattern of scatter is captured as spots on photography film and analyzed. It is also the technique used by Rosalind Franklin in order to prove that the DNA, or the DNA rather, is a double helix structure. Watson and Crick model formulated by James Watson and Francis Crick in 1953. The DNA is a double helix coiled around a central axis, and the chains are anti-parallel with each other. Both chains are 3.4 angstrom apart, and the specificity of base pairing is adenine to thymine and cytosine to guanine, and each complete turn of the helix is 34 angstrom. Alternating minor and major groups are apparent along the axis. Overall diameter of 20 angstrom, and the mode of replication is semi-conservative.
So there are actually different forms of DNA. And let's start with bDNA, which is the most common right-handed helix. So in bDNA, it is seen under aqueous low salt conditions. It is biologically significant conformation that exists under normal physiological condition. And its helix width is 2 nanometers. With aDNA, it is present under high salt or dehydration condition. It is slightly more compact with 11 base pairs, and its diameter is 23 angstrom, which is 2.3 nanometers. So it is also a right-handed helix, but it's shorter, and its orientation is somewhat tilted and displaced laterally in relation to its axis. And the next one is zDNA, which is discovered by Andrew Wang and Alexander Rich and their colleagues in 1979. So zDNA is a left-handed helix. It has 18 angstrom in diameter, which is 1.8 in nanometers, and it contains 12 base pairs. So it is assumed to have a zigzag conformation of backbone, and its major groove is nearly eliminated. Denaturation and renaturation form the basis for molecular hybridization. Complementary nucleic acid strands can reform duplexes. RNA hybridizes with DNA segment from which it was transcribed. And so, in here, DNA's unique hydrogen bonds contribute to its structure, and when it's heated, the DNA double helix denatures and unwinds. The melting temperature represents the midpoint where 50% of strands have unwound. And high melting temperature indicates more cytosine and guanine base pairs that has three hydrogen bonds compared to adenine to thymine pairs that has two hydrogen bonds. In nucleic acid, electrophoresis molecules such as DNA or RNA are placed on a gel. So the negative charged nucleic acid moves towards the positive charged anode under influence of electric field. And the gel spores separate the molecules based on their size, allowing different size fragments to migrate at different rates. Agarose gel with various spore size are commonly used for this, and it can distinguish polynucleotides that differ just by one nucleotide in length. So in blotting techniques, such as southern blots and northern blots, use electrophoresis to detect specific nucleic acid sequence. And
Another is DNA sequencing methods that also rely on electrophoresis.
For the first problem, we have 170 angstrom long segment of DNA molecule has 15 cytosine. What will be the number of adenine in the same segment? To solve this problem, we must first know the given of 15 cytosine guanine in 170 angstrom since the value of cytosine is the same with the value of guanine. So the unknown in this problem is the adenine in 170 angstrom, and all we have to do is multiply the 170 angstrom to one base pair and divided by 3.4 angstrom since we know that the distance of the bases are 3.4 apart, and if we multiply and divide, we will get 50 base pairs, and these 50 base pairs will be subtracted to the 15, which is the value of cytosine, and we will get 35. Therefore, the number of adenine in the same segment is 35. For the second problem, we have how many phosphodiester bonds are present in 80 base pairs. As we did from the previous problem, we must note first the given, which is the 80 base pairs, and the unknown is the number of phosphodiester bonds in 80 base pairs. In this problem, we are going to utilize 2n minus 2, where n is the number of base pairs, and we just have to substitute 80 to the n, which would be 2 times 80, which is 160, minus 2 is equal to 158. Therefore, there are 158 phosphodiester bonds present in the 80 base pairs. For the third problem, we have how many base pairs are present in a DNA molecule with 160 phosphodiester bonds. Given, we have 160 phosphodiester bonds, and the unknown is the number of base pairs in 160 phosphodiester bonds. We will utilize the formula of 2n minus 2 again, wherein the number of phosphodiester bonds is 160, so 2n minus 2 is 160, therefore 160 plus 2 divided by 2 is equals to 81. And therefore, there are 81 base pairs present in a DNA molecule. For the fourth question, we have in a DNA fragment, if there are 8 turns with 40% of the bases are cytosine, what would be the number of hydrogen bonds present in this DNA fragment? So we know that 10 bases exist in each complete turn of the DNA helical structure, and we have to multiply it by 8 since it said here that we have 8 turns, and it will be resulted to 80 nucleotides. Thus, the number of nucleotides, or rather the cytosine, is 40, and we have to multiply it by 80, which will be 3,200, and divided by 100 is equals to 32. And since the cytosine is complementary to guanine with triple bond, the number of hydrogen bond is 96. As we proceed, the 80 nucleotides, we have to minus it to 64, which came from 32 plus 32 of the cytosine and guanine, and we get 16. Therefore, the other nucleotides, adenine and thymine, is 16. Since they are double bond, hydrogen bond is 32. And as we sum up everything, the total number of hydrogen bond is 96 plus 32 is equal to 128.
In this question, given that the DNA of species A has a mole fraction of G plus C, which is equated to 0.36, what is the mole fraction of A? So we're looking for the mole fraction of A, and 0.36 was given, which was subtracted to 1 in order to find the mole fraction of adenine and thymine. And so 1 minus 0.36 is equated to 0.64. But you have to divide it by 2 because you're only looking for the mole fraction of A. And so the answer for this is 0.32. What is the percentage of guanine and adenine in a DNA sequence that contains 20% cytosine and 30% thymine? So you have to remember Chargaff's rule, and so adenine is bonded with thymine while cytosine is bonded with guanine. And so the answer for this is letter A, which is 20% guanine and 30% adenine.